Okay, so we'll move on now to kind of the second part of your area of study. So looking at the brain. So it depends how your schools have kind of been running things, I guess. And yeah, with 1 and 2, there's a lot more freedom. So in year 11, schools can choose like a decent amount of what they want to do, whereas 3, 4 is a little bit more rigid. But if they are following the study design pretty strictly, your school may have done development kind of in term 1. You may have already started stuff on the brain, or maybe going into it in term 2. But I'm assuming most schools would have probably started stuff on the brain already. So we're looking at our study design dot point here, different approaches over time and understanding the role of the brain. So different approaches over time, we're looking at some of these old approaches that may have seemed really good at the time, but now that we know a bit more about the brain, it's a little bit, interesting. So the heart hypothesis and the brain hypothesis, it was this sort of debate, I suppose. Oh, this should be saying in the brain. But this sort of debate saying, is the heart the center of thinking, or is the brain the center of thinking? And then that kind of leads to the mind-body thing. So this idea of, are our brains and our mind the same thing? Is our body connected to our mind? Are they two separate entities? Stuff like that. And remember, this is, quite a couple years back, couple centuries back even. So a lot was not known. Just like right now, a lot of the brain still isn't known, but back then even less was known. So it's kind of a valid question to ask. Now we know this idea of obviously mental processes being in the brain and things like that. But yeah, it was this idea of, how does the brain interact with the body? Does the body influence the brain? Is the mind in the brain? Is the mind in the body? Is the, mind, or is thinking in the heart versus in the brain? Yeah, so a lot of these questions in terms of kind of understanding what the actual point or the actual function of the brain was. And then phrenology, this is a little bit of a pseudoscience as well. So this idea that the physical characteristics of the skull shows you what someone's personality is like. So if you have a little bump here, or your head is flat here, then, you're a more aggressive person, or you're a more intelligent person, or you're a more relaxed person, stuff like that. So obviously that has been kind of disproved, or this idea that, the bumps in your skull don't really dictate your personality. In terms of actually figuring out how, so figuring out what the brain actually does, there were these certain techniques. So ablation, this is a little bit more of an old school one, as you can imagine. So parts of the brain are basically just taken out or destroyed, and then they figured out like, okay, what function is lost? So I guess pretty clear method of testing, obviously pretty unethical. Electric stimulation is obviously a more modern thing as technology grew. But this idea of, when you stimulate the nerves in this part of the brain, what happens? How does it affect this person functioning? Does their arm move? Do they stop talking? Do they, blink, stuff like that? And then that's how a lot of the kind of understanding of what regions of the brain control, what parts of the body or what functions, how those are kind of found out. And kind of, again, further findings from that is this idea that the brain, obviously we're going to talk about, structures of the brain and, main activities that happen in each part of the brain. But again, as you probably know what I'm going to say, we don't like box things into categories. The brain is just a big mix of everything. This idea of it being holistic. So, this part of the brain is more in control with this certain function, and this part of the brain is more in control of this certain function, but they can both have it's like having your fingers in all the pies. I don't know, whatever the saying is. All parts of the brain can contribute to all different functions. And as we'll learn about as well, when certain parts of the brain are damaged, other parts of the brain can take over for that, damaged region as well. Okay, so split brain experiments. These were done in the setting of epilepsy. So they figured out cutting the corpus callosum. So you have your left hemisphere and your right hemisphere. The corpus callosum basically connects it. So it's like your little highway, your little bridge between the two hemispheres. So in people with severe epilepsy, what they kind of figured out is like, if we cut this, it stops that communication. So then they're less likely to get seizures. But then they also found out like, oh, when we cut this corpus callosum, look at all these kind of effects of what happens when the left hemisphere and right hemisphere can't communicate. So this idea of not being able to talk basically between your right and the left. So the left hemisphere is very responsible for this idea of speech, for talking. So if you have, okay, it's going to be a little confusing. I know you guys will hopefully be aware of the fact that the right side of the body is controlled by the left side of the brain. So if you have things in the left visual field, they're processed in the right hemisphere of the brain. And you won't be able to name these because of the idea that you can't extract information from the left side. I hope that makes sense. So let's say, so I've got like, I don't know, an apple in my left visual field. So it's on the left side of my face. Yeah. So the right hemisphere will kind of process that. So that information will come through and it'll be processing that in the right hemisphere. So the left part of your brain knows that this is an apple or describes it as an apple. So the right hemisphere is saying, okay, I recognize this object. What's it called again? But it is unable to take that information from the left side to the right side. So that's why it can't be named. On the other hand, if you've got something presented in the right visual field, you process that in the left hemisphere. And because the left hemisphere has the names for stuff, you're able to name it. So you're able to say, oh yes, it's an apple. Because the knowledge that this thing that I'm seeing is an apple and actually being able to say that it is an apple, that's in the left side of the brain. So anything that's processed by the right side of the brain, i.e. the stuff on the left side, I know it's complex, that's what you won't be able to name. Because again, that road is cut off. The right side of the brain is by itself. The left side of the brain is by itself. They can't, any functions that are kind of exclusive or mainly on one side or the other, they can't be shared anymore because of this idea of the band of nerves being gone. Okay, so talking about this idea of nerves, this and that, neurotransmission is important to understand this. I will say in the new study design, so for this year, the kind of important, I guess, parts of a neuron and the actual synaptic stuff that's been sort of de-emphasized or emphasized less. But I think it's important in being able to understand what we actually talk about in terms of neurons and synapses and that sort of thing. So I do just want to flag, it is probably not like the highest yield stuff, but I think it's still important for actual understanding. So here is what we call your presynaptic neuron. So this is the synapse. The synapse is the gap between the two neurons. So again, very intuitive in the names. Presynaptic neuron, the neuron before the synapse. Postsynaptic neuron, the neuron after the synapse. So the synapse is interacting between what we call the axon terminals, or the terminal buttons, and the dendrites of the next neuron. So I like to think of it as like the toes and the hair. I know that might sound a little bit strange, but you think about the impulse or the communication going from the top to the bottom. So essentially the dendrites, and it passes through the soma, passes through the axon, through to the axon terminals, which is what we see happening on this side. So to me, what I like to think of it as is like the dendrites are like the hair, the soma, almost like the head, because it's like the cell body, it does all the complex stuff. The axon is the body, and then your little terminals, these kind of look like toes or feet, maybe. So that's how I like to think of it. So it goes from top to toe, top to toe, top to toe. And so you have, 300, let's say, neurons lined up one after the other, top to toe, top to toe, top to toe, top to toe. The information is just shooting from one neuron to the next neuron to the next neuron to the next neuron through these little synapses. That's how it's going from one neuron to the next. So to cut a long story short, you have your little neurotransmitters in here. They're basically released across this gap. and they bind to receptors on the postsynaptic neuron. They do their little thing, and that sends the impulse through this neuron to the next one. That's the simple gist of it, really. So when you form new neural pathways, you're forming new synapses, new connections between different neurons, and obviously kind of all the physiological stuff that goes on in terms of number of neurotransmitters, number of receptors, all that kind of stuff. But yeah, that's sort of the basics of it. So I think understanding that can help you a little bit in your knowledge when it comes to stuff like synaptic pruning and rerouting, that sort of stuff. Okay, in terms of the actual structures of the brain, you're looking at hindbrain, midbrain, forebrain, and the cerebral cortex. So here we have our hypothalamus and our thalamus and our cerebrum. So these are in our forebrains. You can kind of see these in here. Don't worry too much about, the fissure or anything like that. Here's the corpus callosum, sort of for context again. It's just that line. So we look at our hypothalamus and our thalamus, very much to do with sort of regulation. You're thinking about hormones, you're thinking about homeostasis, stuff from, your visceral, your sensory organs. Lots of this kind of filtering of information, regulation, that sort of stuff. Cerebrum we'll look at in a second. Midbrain, you're kind of thinking of your pretty important function. So your reticular formation, stuff to do with consciousness, arousal, that sort of stuff. And then hindbrain. So your medulla is super important, connects your spinal cord to your brain. So if your medulla is injured, it's like really key function. So things like breathing, again, sort of consciousness, this idea of actually connecting the spinal cord, so that sort of highway of neurons, and ultimately the rest of the body through to the brain. The cerebellum you think of with stuff like balance and like coordination. Okay, so this is our cerebral cortex, or our cerebrum. It's basically the outer layer of this brain. And so it's separated into your frontal, your parietal, your temporal, and your occipital. So frontal lobe, as the name suggests, at the front. So here's what we think about when we think of like complex, like actual thinking. So when you think about like, what does the brain do? Like the really basic instinctive stuff you think, that sort of happens in the frontal lobe. So like, I don't know, doing your maths and like reading and stuff like that. Your problem solving, your abstract thinking, all those kind of things. Your primary motor cortex, it's basically like, I'll try and draw it. It's like a little band that sits along here, kind of. And as the name suggests, very involved with your kind of motor stuff. So motor movement, voluntary movement, that sort of stuff. Your parietal lobe, so your somatosensory cortex basically is like touching the primary motor cortex, and it sits here. So at the front, essentially, of the parietal lobe, and that's involved with a lot of sensory stuff. So kind of sensing, information like touch or heat, things like that to do with sensory stuff. So somato in terms of the body and sensory. Occipital lobe, basically to do with eyes. So everything with vision, as the name suggests again. Temporal lobe, so kind of sits on the side, to do with memory, but also particularly to do with hearing. So you've got your primary auditory cortex. So you can see, again, motor cortex, somatosensory cortex, visual cortex, auditory cortex. They tell you what the lobes kind of do. So yeah, we think about to do with hearing. Also involved with memory, as I mentioned, you've got some important structures in there to do with memory and stuff with like facial recognition as well. Okay, so these are two super important areas to be aware of. So you've got Wernicke's area and Broca's area in the left hemisphere. So remember we talked about that corpus callosum and how the left hemisphere is important for speech and language. So Wernicke's area is in the temporal lobe, so here, and Broca's area is in the frontal lobe. Remember that. Something you also have to remember is what they actually do. So Wernicke's area, the way I like to remember it, is temporal lobe is with auditory. So Wernicke's area is to do with actual comprehension. So understanding what words mean. Whereas Broca's area is to do with speech production. So you can kind of think about like, the motor cortex, actually like maybe moving the mouth, moving the muscles and stuff like that, that can kind of help you distinguish between what they do. So if you have a question and it's like, this individual is struggling to understand language. So what that means is you've got a damaged Wernicke's area. They will be able to speak very clearly. Their enunciation will be fine, but it will be completely jumbled. So it'll be like, cat, spanner, computer, wall, light bulb, right? So it doesn't make sense, but the actual production of the speech is fine. It's clear. Whereas if you have damage to Broca's area, they will be able to fully comprehend what you're saying and kind of formulate that sentence, but they won't be able to actually speak it. So it'll just be complete gibberish in terms of what they're saying. So you can see how these two areas are really, really important in terms of actual speech and language. Okay, so we now look at the idea of how the brain changes and how, when we experience things, when we learn things, how that actually changes the brain physiologically. So we look at these three kind of processes: myelination, synaptogenesis, and synaptic pruning. So myelination, you might be familiar with the concept of myelin that happens sort of on the outside of your axons, and it's very important for sort of insulation. So just like an electrical cord, that's got its little insular tube. It's the same thing for a neuron. It just helps increase the efficiency of the electrical impulse, and it prevents sort of interference from other neurons as well. Synaptogenesis, again, as the name suggests, I feel like I'm saying that a lot, as the name suggests, this kind of creation of new synapses. So that occurs really rapidly after birth. So when you are a baby, like babies' heads are huge in proportion to their body. Their brain's just growing like crazy. So you have a lot of these new synapses, these new connections that are forming, as the baby like learns a lot of stuff as well. Synaptic pruning. So this is important. This happens again kind of after this period of synaptogenesis, when the brain realizes, okay, I've made all these lovely pathways, all these lovely new synapses, all these connections. I'm not using them. I'm going to get rid of them. Again, just focusing on, efficiency and keeping resources that the brain needs. So if the brain has a pathway and the brain realizes, I haven't used this pathway in a long time, I'm going to eliminate it. I'm going to get rid of this because it's unnecessary. So that's that idea there. Super important concept to understand now is this thing of plasticity. So when we think about, synaptic plasticity, neural plasticity, it's this idea of being plastic, really. So being flexible, changing over time. So as you experience things, as you learn things in particular. So again, this is something you'll go over next year in 3-4 to do with learning and memory, how the brain and like at a synaptic level, how it actually changes as you learn information. So adaptive plasticity, this means, again, as the name suggests, adaptive. You're adapting for something. So you're compensating. So if you have a certain region of the brain that perhaps is damaged, the brain adapts and kind of other areas of the brain essentially overtake or take on that role. So that function basically isn't lost. So if you've got a huge chunk of the brain, let's say a huge chunk of the right side of the brain, so maybe a lot of your left-sided, limb functions are lost. If you're a pretty young person, the body may be able to actually adapt. So a lot of, rehab and physical therapy, you may gain that function back. Compared to an older person whose brain is basically less plastic. They have this, they have a lower ability for the neurons to be able to actually be as adaptive. And that kind of links to this concept of developmental plasticity, where we're kind of seeing that, in a baby, this development where we've got stuff like synaptogenesis. So this kind of creation of new pathways rather than trying to get established pathways to take over functions. Okay, so we have these two important processes, so rerouting and sprouting. It's in the name. So rerouting, we're just thinking of when you have a healthy neuron and perhaps another connection has been damaged or another pathway has been damaged, the neuron will just take another path. So it's basically like, you've got a path, it's blocked, road closed, you've got roadworks, you just take another path, you reroute that. Sprouting is when you've got this neuron and it basically just sprouts like a little plant. It just grows these extra networks, so extra dendrites. Remember, that's where your receptors are. And so that means it can just connect and hold hands with a lot more neurons. So now you've got a lot more connections in that way. So again, that's kind of what we've talked about before. So this idea of rerouting and sprouting, it allows the brain to, again, adapt and just take over and replace lost connections. Okay, so if we're thinking about brain injury, again, this is a very broad dot point. So you're thinking about how somebody's brain is damaged based on, or how a person's functioning is affected based on damage. So again, your biopsychosocial kind of approach. So you can see there's a long list here. There's a lot of other ones that are involved as well. It just depends. It really, really depends where that brain damage is, the extent of the brain damage, how old the brain damage is, really, all that kind of stuff. So you can see a lot of things: reduced motor activity, reduced planning, personality changes. Again, think about your lobes. Think about if a brain injury was in your occipital lobe versus your frontal lobe, what the kind of damage might be there. Okay, again, sort of like a bit of a wishy-washy dot point. The contribution of contemporary research to the understanding of neurological disorders. So the example one that we'll talk about in this lecture is Parkinson's disease, but you can imagine there's a lot of other neurological disorders, Alzheimer's, for example. And what the point of this study design dot point is to have you be aware of how contemporary, so modern research, recent research, allows us to understand this sort of stuff. Because again, be mindful, a lot of the brain is unknown. Like even right now, a lot of the brain is known, but there's still so much that is unknown, and it can be very hard. It's a hard thing to kind of figure out what the brain is doing. So that's why it's this idea of kind of modern and recent research, how that's kind of helped us in terms of, diagnosing and understanding things like Parkinson's, things like Alzheimer's, other forms of dementia as well. So with Parkinson's, the basic idea is that you have neurodegeneration. So you have neurons in this area called the substantia nigra, and they basically just die. These neurons are responsible for producing a certain hormone or a certain neurotransmitter called dopamine. So logically, these neurons are dying. These neurons are meant to be producing dopamine. You're going to get low dopamine. And dopamine is important in your primary motor cortex, so what we talked about here, in terms of your initiating and controlling voluntary movements. So if you've got that impaired communication there, you're going to get impaired control of those movements. So that's why you often see with Parkinson's, a big symptom is tremor. This idea of controlling these voluntary movements is affected. So then you see that tremor, and you see, other things, perhaps to do with gait or posture as well. Lots of things to do with that impaired control of those voluntary or, skeletal muscle movements. So in terms of some contemporary research and how this allows us to understand it, animal studies, this is one way that you're kind of looking at understanding Parkinson's. So as I mentioned, it's very hard to figure out what's going on in the brain because everybody that has a brain is living and they're a person. And so it's hard to be like, oh, let's experiment on you. Let's use you as our little guinea pig. So especially in the past, now it is, I guess it's a lot more controlled. But animal studies were a big thing that were used. So in animals, typically like rats and things like that, inducing Parkinson's in them and then understanding how certain medications might work, understanding how Parkinson's is affecting the brain, and then assuming that that's how it happens in a human as well. Neuroimaging techniques, these are a really big one, obviously very, very, very helpful because we're looking at imaging, the brain in which the, disease or disorder is actually happening. So, things like MRI, a PET scan as well, even like CT scans, other stuff like that, is really helpful in determining what's going on in an individual's brain when they're alive and functioning. So if you're looking at, Parkinson's, you can look at what's going on in this person who doesn't have Parkinson's versus what's going on in this person who does have Parkinson's, and noting, okay, this part of the brain has shrunk. These neurons are hyperactive. These neurons are hypoactive, stuff like that, and determining what's kind of going on there. Okay, and I think this is the last dot point. So CTE. So again, this is a new dot point for this study design, but looking at chronic traumatic encephalopathy, again, as an example of emerging research, this idea of contemporary, recent, new into progressive and fatal brain disease. So CTE is basically, again, it's a pretty new thing, hence the emerging research, or like the knowledge of it is fairly new. And you see it a lot with, I remember watching a documentary about this when I was in year 10, when I was doing 1-2 psych. And it's about like, NFL players. You probably would be aware, or, if you've heard of CTE, you probably hear it in that context. So NFL players or other kind of contact sports, perhaps like rugby, things like that. A lot of these retired players are developing this disease, this brain disease, which is CTE. So encephalopathy is basically just this idea of, kind of impaired neurological functioning. And so chronic is over a long time, and traumatic. So this kind of traumatic source of injury. So the trauma is coming from concussions. So those repeated hits, in a contact sport, that's that trauma. And chronic means over and over and over and over again. So if you've got one really bad concussion that happens and you develop, a brain disorder or encephalopathy, it's not CTE because that's come from one really awful kind of event. Whereas chronic, it's happened over a long period of time. So this chronic trauma has resulted in this neurological decline, basically. So in terms of your symptoms, you basically result in this dementia. So again, you can kind of associate things like Alzheimer's or, dementia. You're seeing a lot of memory loss, personality changes, mental health changes, cognitive changes as well, that you kind of associate with dementia. And with this idea of CTE, they were seeing this in obviously retired players, but who are of a fairly young age as well. And ultimately, as it is a fatal brain disease, again, dying at a young age. So then kind of you usually see dementia and Alzheimer's, the onset being, in the elderly. So seeing this in relatively young people as well. In terms of diagnosis, so again, it's an area of emerging research. Not a lot is known about it. And just like Alzheimer's, you can only diagnose it post-mortem. So once you actually look at the physical brain, that's when you can diagnose it. So in people with Alzheimer's, for example, when you diagnose them with Alzheimer's, you're not 100% sure they have Alzheimer's. You're thinking, based on the history, based on this imaging, based on what they're telling me, based on perhaps how they respond to medication, I think this is Alzheimer's, but you can only definitively diagnose it post-mortem. So again, that's kind of the idea here. So based on their history, they played contact sports for a long time, they got concussed a billion times, they're showing all these signs of CTE, of dementia, then, it's probably CTE. Again, you can use imaging as sort of a backup, but it might not be the best technique to diagnose it. In terms of treatments, again, really, really hard. It's in an area of emerging research, so not a lot is known about it, therefore not a lot can be done to treat it. So a lot of it is like supportive or palliative, so kind of aimed at decreasing symptoms, but not at the actual onset of the disease. So it's not curative. It's more so, helping with the anxiety or the memory loss and stuff like that. Okay, hopefully that is all making sense. Again, feel free to ask questions if you have any. We'll look at some more practice questions before we go into scientific skills. Okay, so the term adaptive plasticity refers to changes in the brain in size, a newborn baby, response to hormones, or response to learning and experience. So based on the things we've just talked about, it's not size. We know plasticity is more so the kind of actual connection. So we're not thinking changes in the brain in size, a newborn baby, again, we're talking about how adaptive plasticity, not developmental plasticity, which is probably more what you'd see in a newborn baby, but adaptive is, again, if you've got brain damage, kind of overcoming that and changing roles, certain pathways in the brain, or as you learn things, as you experience things, changing, again, neural pathways, you're rerouting, you're sprouting. And then response to hormones, again, not really the best option there. D is the answer that we're looking at. Okay, imaging technologies have been helpful to researchers in the identification of localized changes of the brain as a result of learning specific tasks. This is because researchers are able to observe changes to the structure of the brain, observe a live brain during the learning process, perform surgery to the brain as the person is learning, compare and observe changes to the brain before, during, and after learning. So again, we're thinking about the identification of localized changes of the brain as a result of learning specific tasks. So it's very important for this, again, because the answers are relatively similar, to think about the actual context. So observe changes to the structure of the brain. Potentially, if you look at D, compare and observe changes to the brain before, during, and after learning, very similar. A is just a little bit more vague. So you'd probably be flagging those two. Observe a live brain during the learning process, not like necessarily the best option in terms of thinking about imaging technologies, or not the best way to describe it as well. Perform a surgery to the brain. So imaging is not surgery, you're not opening the brain. So then again, you're kind of at D. So this is a good example of when it's the best answer, not necessarily the right answer, because A is like, you'd almost think about A being the right answer as well. But D is just a little bit more specific and answers it a little bit more clearly. So you're comparing and observing. So if you're identifying localized changes of the brain as a result of these tasks, observing it before, observing it during, and after the learning, that gives you a really good picture of what these changes are, as opposed to observing change to the structure of the brain. I mean, technically, but it's just a little bit more vague. So that's why we're going with D. I do want to flag this though while we're talking about this. Do not necessarily think, and I guess this isn't a good example of that, but if you get a question and the answer is vague, don't think like, okay, it could never be this question. All the time, and I mean like all the time, this is almost like more rare. The vague answer is often the right answer. So you might get 3 answers that are super duper specific, and they'll be talking about processes in really big detail. They'll use all this terminology that's relevant to the topic. And then one will just be very general. And you'll think to yourself, oh, okay, I think this general one, like, technically is right, but it's just so non-specific. It's so vague, so it must not be true. Do not think that way. If the other 3 are wrong, then they're wrong. And often it's because they're too specific. So again, this is a bad example of a question like that, but you may have one, answer where it's a bit vague, but like it's generally right. And then the, B, C, and D, they might be super specific, but there might be one word in there that makes it incorrect. So don't kind of fall into that trap of thinking just because this answer is vague is probably not true. But in this circumstance, it was too vague, so it wasn't true. Okay, so a short answer question here. Stan, who was 3 years old, and James, who was 33, both sustained head injuries and suffered damage to the same area of their brain. As a result of this damage, both have difficulty speaking fluently and moving their right arm freely. So, name the lobe and hemisphere of the brain that have most likely been damaged. And then part B, with reference to types of brain plasticity, explain why Stan is more likely to have a faster and more complete recovery than James. So, something else with psych in your short answer questions, always mention their names. Always. Assessors and your teachers will literally make you lose a mark because you haven't mentioned their name. And again, which is why I always say psych is so much of application. All application. So to show that you're actually applying it to the scenario, they like to see you use their first names. So bear that in mind when you're answering part B. Okay, so in terms of the answers that we're thinking of, so you can see, name the lobe and hemisphere. So hemisphere, you're thinking about right or left. So it said to you that both have difficulty moving their right arm freely. Remember, we've talked about our hemispheres. They control the, what we call like the contralateral side, so the opposite side of the body. So if your right arm is damaged, your left side of the brain controls your right arm, or the right side of your body. So you know that the hemisphere will be on the left. Then comes naming the lobe. So speaking fluently. So what have we talked about in terms of where speech is? Speaking fluently, we're thinking of the left frontal lobe there. So you can see that those are two aspects of this question, but it's 1 mark. So if you were to get one of them wrong, you'd get 0 marks. So there's no half marks in Psych, 3, 4 essentially. And so often teachers will replicate that. So I think when I was in year 11 or doing my 1-2 Psych in year 10, we didn't do half marks. You got 0 marks or 1 mark because that's how VCA does it. So with reference to types of brain plasticity, so specifically for this one, I hope you guys were thinking of mentioning adaptive plasticity. You would explain why Stan, who is 3 years old, is more likely to have a faster recovery and also a complete recovery compared to James, who's 33. So you bring in their age and this idea that the younger you are, the more successful the brain is at kind of adapting and forming new circuits and things like that. And how that will happen faster, again, because you're younger, but also complete. So in, let's say that the, right frontal lobe was removed from a 3-year-old and a 33-year-old. In the 3-year-old, it's likely that they will actually be able to kind of function, let's say, like as normal, and there won't be any major deficits noted. Whereas in the 33-year-old, they may be able to regain a decent amount of function, but there will still be part that's completely lost their function. So that's the idea of the complete recovery. So you can see here, name the lobe in the hemisphere. So the left frontal lobe, blah, 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 controlling movement of the right side of the body. So the left frontal lobe, this area, hopefully you're thinking of Broca's area. And again, that's how I kind of think of it, like Broca's area, motor, like actual production, and then auditory cortex, Wernicke's area. So that idea of hearing and understanding. Okay, so 6B, blah, 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 blah. Correct response stated that. So Stan's 3-year-old brain has the benefit of developmental plasticity or access to both developmental and adaptive. James' mature brain will be limited in its recovery. So hopefully, like, again, this is obviously you're thinking of like a very well-versed student with VCAR marking here. I know you guys we're just talking about this. So hopefully what you guys are thinking about in that time is this second dot point. So James' mature brain will be limited in its recovery to the mechanism of adaptive plasticity. And thinking about this as well, the processes of rerouting, and sprouting in adaptive plasticity are less efficient than the ability to form new circuits. So it's just this idea that when you're 3, you may have these kind of aspects of synaptogenesis in which new synapses are forming. Whereas if you're 33, you don't really have new synapses forming. It's more so, again, the rerouting and the sprouting as well.